So hi everybody. Thanks for the intro. And yeah, so for this talk, I kind of wanted to be more like a discussion. Like, there's so many tools like AlphaFold 3 or DiffDoc that you might have heard of, and you kind of don't know where to start. So I think this will be pretty helpful for like trying to pick a project or pick a tool that you might want to play with, because each part of the paper that I'm presenting is essentially like modular kind of code block that you could play with on like a notebook or something. So, but yeah, so I'm a fifth year PhD student in Charlie's lab, the Charlie Brooks. We do mostly uh, classical mechanics or stat mech molecular dynamics type stuff. So that's where a lot of the inspiration for the molecular mechanics part of this talk is from. And then I'm also interested in machine learning methods. So that's why that's like bridged together. And yeah, thank you for the opportunity for the DCM Tools and Tech seminar. Check out the YouTube channel. They have lots of cool videos. So mine will be up there hopefully. Um, so yeah, so same summary. There's like way too many tools. Like if I ask you like, hey, how do I run AlphaFold 3? And you're like, I really don't know. I wish I had like like a little 100-line piece of code that I could follow and be like, okay, yeah, I could read the code, because whenever you go through papers and like especially machine learning or like tool papers and it's like full of math, I just can't read it. So there's no math in these slides. It's just concepts and code, which I think is more digestible. Yeah, so my paper, I guess why I'm doing this talk is it was published in Bioinformatics earlier in January. So there's the associated article and GitHub repo with the paper. But so the next thing, just I'm going to be breaking this talk into like two parts. So first, we're going to start with concepts, because sometimes you say stuff like, oh, I used AlphaFold 3, Doc did, binding affinity, and you're like, I don't know what any of those actually do on their own. So I'm going to be breaking down the individual concepts, why I linked together like specific things the way I did. And hopefully it makes more sense that way when we actually like look at the code and you could be like, oh, that's the step he was talking about. So first thing, proteins. So proteins, you can represent them as a sequence, as a structure, as the whatever biological function that they do. Uh, the problem is all of those are not um, low dimensional spaces. Like any protein sequence is n choose 20. So a 400 amino acid protein is 400 choose 20 different possible combinations, which is too much. And then structure, that's continuous in Cartesian coordinates, so it's kind of like you can't even make it discrete. And then function, there's thousands of classifications. So proteins literally do everything, which is why I like studying them, because they're part of making your cells, move. They're why ATP is used everywhere, because every protein loves ATP to do some really cool activity. And the protein that I'm going to be talking about is in, oh, hey, Chad. So this actually, my experimental collaborator, he's actually half half the stuff I talk about, he's like involved in it, mostly on the wet lab side. So that's great. All right, Chad, you want to introduce yourself? No, no. Okay, he's a fifth year from Allison's group. So if you're interested in experimental side of this, because this is also proteins are just not computational. Like we study them in the lab, and that's what Allison Ryan's group does. And I think Chad is like a good representative. He's a fifth year like me, so lots of wisdom. So all right. So the point is, I guess what they actually do in a collaboration with my lab and Allison's lab, what that typically looks like is, if you want to explore protein sequence structure space, is you start with the natural protein, like maybe one crystal structure that you're pretty confident about. You do some docking MD, and you're like, okay, these residues might do something, and then you test it experimentally. And that's kind of how it mostly stays. Like, so for rational design, they do crystal structures in molecular dynamics and QM, which is expensive and only, you can target the single structure. And most of the time, you can only really understand the binding site. You can only really see like, hey, these residues interact, but I don't really know what they do. I just know they interact with the ligand, which is not often that useful. So I'm going to be talking about designing a workflow for navigating this really high dimensional space by combining tools that are well established for multiple fields. So all of these tools individually, they deserve their own time to be talked about because they've been built on like, 20, like all the molecular mechanics stuff is at least 50 years of stuff that it's built on. So I'm just going to be covering the part of why you should use it. So it's just going to be the code and what you can actually do. So I guess to make the example simple, we're going to do, I'm going to pretend like I'm in a collaboration with our lab, what a normal, typical computational biological or chemistry lab looks like. We have a biocatalyst. It's like a 400 amino acid enzyme. So lots of possible sequence combinations. You only have a handful of crystal structures, but you tested them in the lab and you want to go to your computational collaborator and say, hey, this is the data I have, and this is the data that we have for a lot of proteins because proteins have been engineered for a long time. Is there like a smart way I could plug all this data together and get something meaningful out? And yeah, so that's the whole point is to work with like a realistic data set. So right now, if, you know, Chad was engineering a protein, he would want me to give him a model that he gives a sequence. I tell him what the function of that sequence does so he can synthesize it or whatever. Now, obviously, that's impossible. So we're gonna insert a little bit of stuff in the middle. So sequence space 400 choose 20, way too big. We can look at real enzymes and see, you know, you don't have to search random sequences. You can search sequences in the same enzyme family. And then you can link that to function. So that's pretty standard. Next thing is, okay, well, how do you go from MSA to function? So you can do some analysis with your eyes. Like you can open up the MSA, look for columns, and then your eyes are like kind of struggling. But that's only good for binding site residues most of the time because there's just way too much stuff to look at. So let's just say there's a helpful ML model that does that job for you. So takes the MSA, does something, spits out the function that Chad wants. And we're going to talk about how it works, but you could pretend like each column is a feature. So you have like an Excel spreadsheet with your MSA with all the gaps and residue names, and then you just fit it like you could fit it with a linear model. I'm just using a random forest because it's slightly more complicated. So the problem is, in a realistic setting, there's no ML model you can train with like 10 to 100 data points that can map a multiple sequence alignment to a function. So we're going to pretend, just like if we keep going hypothetically, let's say this model is trained on some synthetic data. So this is actually pretty common. So if you think about it, like if you want to train a large language model, it might be easier just to take the output of GPT-4 and train your model on that. So that's synthetic data. There's also weak supervision where you have another model that weakly labels every point, and then you fit a model on that, and it learns to somehow map those weak labels to the particular space you're working in. So this way, the labels might be from literally anything. It might be some complicated, dynamics analysis, QMMM. But if I just turn it into a label, then it's a really easy problem to map. And then if you pick the label smartly, like if it's based on some structural or domain knowledge, you can train a model like this. So to get that kind of domain knowledge, so that's the point of molecular mechanics is, people have been using structure prediction, docking to engineer proteins for, I would say, at least 30, maybe 40 years. And modern force fields in like charm or OpenMM are pretty accurate. So I think the problem now is we have a very well established, well, QMMM is very well established, molecular dynamics very well established, but it's hard to connect it and see how it would be useful for training a machine learning model. So that's the point of this paper here is that, hey, for every sequence here, we can get the structure pretty simply just for an AlphaFold. And then, which sounds hard, but then you could see in the code snippets, oh, it's actually, you just run AlphaFold, and it's not that intense. So do AlphaFold, use a molecular mechanics force field to put the cofactor for this protein that I'm working with, and then do some docking and refinement in the force field. And that's basically condensed into just saying, hey, structure, doc, give me function, or give me a label. And the label doesn't have to be perfect. It just has to capture some pattern that is repeating over and over and over again in like thousands of sequences. So if I told you, hey, my MSA is like 20 sequences and I have predictions for and labels for all of them, and I train a model on that, you'd be like, no, that's ridiculous. But let's say you have 1,000 data points and you did 1,000 docking, 1,000 structure predictions, 1,000 refinements, and you're like, hey, I trained a model on these labels. Now, obviously, the force field is not accurate, perfectly accurate. There might be specific side chains that don't actually look, be in that dihedral space, and the ligand doesn't actually prefer that the minimum energy posed in that force field or something. But nowadays, I think modern force fields are good enough to cover that. So, okay, pretend like MSA, something is labeling all of these MSA sequences. You could train a normal model on it. And then you're like, well, and then Chad's like, I really don't want to plug in thousands of sequence combinations. Can you just tell me which residues you want to mutate? Which is the biggest request from an experimentalist collaborator, because that's all they care about, right? Yeah. Um, so yeah, they don't care about the hard work. They just want, hey, which residue do I mutate and why? And since we have a model, explainable AI has also been around for like, I would say 10 years, like Lime, SHAP. Um, there's like a new one that just came out, but they're all pretty well established, and this isn't a complicated model, it's just a random forest. So it's pretty interpretable. So like if you think about how a decision tree works, we're going to look at this MSA column, position 239, which is the same residue aligned across all of the sequences slash protein. And then we're going to say, if it's F, go this way. If it's Y, go that way. And then if you do that enough, you could just basically break down any problem into like, hey, can you plot how any particular residue affects the final function that you predict? And that makes it actually really easy to interpret, which is, I think, the most fun thing, because sometimes it's hard to understand why you're doing this, but having a nice plot at the end, like of different residues and their importances and their kind of their impact on different aspects of the function of the protein is a really useful thing to tell your experimental collaborators. So that was a simplified version, the last 10 slides. Basically, the paper is a little more complicated, but the concepts are the exact same. I didn't like skip over anything. We have the MSA, we do some structure prediction AlphaFold. Now we just have a bit more details on how I dock it, and I'm specifically predicting stereochemistry and reactivity for these enzymes. That's the data that we have. And because stereochemistry is typically a structure, like you can infer the stereochemistry from the structure of the protein because you can dock different enantiomers, you can dock the substrate, see which the facial selectivity, and then you can assign some score that says, hey, enzyme prefers that. We do the same with activity. We know that in this protein, it interacts with an arginine really strongly. And if you mutate that arginine, it kills enzyme. So I trained another model with some distances and some, the docking energy and the predicted binding affinity from some other model. So it's like a consensus score, labels it reactive or unreactive. And yeah, so if you're interested, particularly like in the major kind of program or concept or tool behind each of these steps. They're laid out, and they're laid out in the following slides, but I think this is like a good summary. Like if you want to learn about AlphaFold 2, then check out that slide. Or if you want to understand how rigid receptor docking, you could do it really fast with like fast Fourier transforms. You could check that out. But I think each of these individually is like too long to talk about. So I'm just going to show you the code to how to run any of these. Yeah, so that's the point of the rest of the presentation. So I only have like 30 slides and it's 12:17. So, I think this is the part of the presentation that you should ask like, hey, how do I run that? And like, can I run that with this particular, example that I have or this particular um, toy that I want problem I want to teach myself. Like if I want to teach myself how AlphaFold 2 works, can I run your notebook with this? And I think most of the time the answer is yes, because the notebook you need basically two different content environments, one for AlphaFold, one for everything else. AlphaFold is kind of the environment's very big for it, so I couldn't manage the dependencies. So just two content environments, Jupyter Lab, spin it up. You won't be able to run the examples with GPUs, but you can run CPU and mostly fine, and they should work perfectly fine. But to start with, we're gonna talk about specifically what enzyme are we talking about. So this is a flavin-dependent monooxygenase. So what that means is we can start here. Flavin, that's the FAD cofactor. So we're going to have to model that into the protein because AlphaFold only predicts the apostructure without any cofactors bound. Now, AlphaFold 3, you could plug this in and specify I want FAD, but it doesn't give you a lot of control. And if you want to do 1,000 at once, it's, you can't sit on the server and do that all day. So but why this system specifically is because the substrate, we actually know a lot about the substrate. So the substrate sits in the pocket and it has a particular phase that is exposed to FAD, and FAD takes an oxygen from or O2 molecule, interacts with it, then shoves the hydroxyl group onto a particular phase. So that's what happens here. That's why this is the substrate, and then it gets a hydroxyl group, and this one is sticking out of the plane. So I think this one is like the R enantiomer. And then if this ligand was flipped around or in some other orientation, but with the other face somehow exposed to FAD, then you'd be like, hey, I could predict the stereochemistry. I don't know if I'll be reasonably accurate, but it'll be decent. It's better than nothing. And for reactivity, we have previous site-directed mutagenesis experiments with tyrosine 239 and 206, and we know that from a previous paper that Chad got a paper in PNAS, and that's why I really like Chad because I was dragged onto it for the computational modeling. So that's why I could say I have a PNAS paper. But basically the point is in this paper we wanted to understand why exactly a specific enzyme prefers like an RS enantiomer, because if you're thinking about it, why do we care about this reaction? It's used for downstream synthesis of organic natural products, or not organic, biocatalytic, what are they called? Natural product synthesis. Yeah. But a lot of natural products are basically antimicrobial. They're potential drug compounds. A lot of current FDA-approved drugs are from just some random natural product that somebody picked from a tree, and it turned out to be a cancer drug. So, but the thing about drugs is the stereochemistry is super important. Like, I think we've all heard of there's like examples of drugs that the other enantiomer will kill you, the other enantiomer will actually, I guess, take away your anxiety. I don't know. It's not that like dramatic, but stuff like that does happen. So we have to be basically almost if we can make any particular enantiomer with 100% selectivity, that would be pretty helpful for different substrates. So that's why we're studying this system. So in the oops. So in the PNAS paper, what they did was it's kind of like a fancier version of building an MSA. So built an MSA, and then we can connect the real enzymes with ancestral proteins. So these ancestral proteins kind of are chimeras and kind of try to explain how each of these extant enzymes evolved. So you like build a phylogenetic tree, and then each ancestor is like a mishmash of all these different uh, enzymes before it. So that's really helpful because the ASR does the work of, hey, these enzymes have specific mutations which makes them related. And related enzymes typically have the same function. So you could basically block different sequences of ancestors and extant enzymes. And that's what he did. He basically sampled a bunch of sequences from the ancestral tree, and he tested them with 4 different substrates. So we have about 60 enzymes with cell lysate, and we know this enzyme kind of does S, kind of does R, which we'll talk about the data more, but that's why we're studying the system, and that's kind of a realistic amount of data that you're working with, because you didn't even purify the enzyme, so it's just cell lysate, which is actually not that hard to do 60 cell lysate reactions. The hardest part is just getting the sequences, but since we have ancestors, we're getting sequences that are kind of more informationally rich, which is why because we've modeled this system and because the sequence space holds so much information, that's why I wanted to study this particular system, because the point is I want to be able to validate, hey, I do find that these residues are important or control stereochemistry the same way Chad did. But, despite all of this fancy stuff, we only know what a handful of binding site residues do. I think we only know what, like 6 or 7 kind of. You tested like 10. And then, if you enzymes are much more complicated than 10 binding site residues. Like there's second sphere mutations that can somehow affect the structure of the binding site, or maybe there's like a particular secondary structure element that's evolved in 2 different enzymes or enzyme families, and that somehow modulates the dynamics or something. So enzymes are super complicated, and just knowing the binding site is just not enough to do engineering, which is why directed evolution, you usually do an expanded shell, about like 90 residues, or you could do error-prone PCR and get random mutations scattered around. But it's not enough information despite how well studied this system is. So that's why it's a great model system. Yeah, so just to, I guess, get more into data. So this is the ancestral tree that Chad built with Troy Wymore from Stony Brook. Shout out. Each point in here is an ancestor, and these sequences here are the extant sequences. So the diamonds are extant. So this one would be like a hit from like BLAST. Like you'd go to NCBI BLAST and get the MSA, and then all of these are hits for like a particular entry sequence. We picked one, or Chad picked one sequence that we have a crystal structure for, and got all of these hits from BLAST. And then the ancestors are built to kind of be chimeras of these sequences. So like, for example, maybe this. So like, if we zoom into this ancestor, or over here. So these regions are colored in because these are the particular clades that Chad studied, because there's a lot of enzymes that actually don't do this reaction, but they're somehow structurally related. So, but basically what happens is the yellow region is one particular extant sequence that we're interested in, because there is there's a sequence, right? And all these yellow dots mean these enzymes do S stereochemistry with the substrates that Chad tested. And then because we can do ancestral sequence reconstruction, we can actually just walk up and see, well, what ancestor, changed to make the other plate over here do R stereochemistry. So you can walk up the history, and then these ones will carry mutations that kind of try to explain why those sequences diverge. So that's why it's a great kind of training set to try out. So, and then the numbers here is Chad assayed 67 total enzymes with 4 substrates. So that's 67 times 4 data points with cell lysate, which is pretty standard. And we have a handful of R, handful of S. And in the paper, Chad showed that there's one key binding site residue that is, if you look at the MSA column between these groups of sequences and the ones with the opposite stereochemistry, there's a pretty clear marker in the MSA that, hey, phenylalanine, the tyrosine makes it go from S to R. So that was like basically the whole point of the paper. And then we talked about some other binding site residues somewhere in some distant clades, which is why this particular clade is highlighted. But the major point here is that we have all of these unlabeled data points in purple, and we can help flesh out the missing gaps. We can also help figure out, hey, these are totally unknown clades. Is it worth our time looking in these? Are there any particular interesting features that maybe they don't even do the reaction, but the way the ligand is positioned in that pocket would be helpful information, like different residue combinations we can try? Because technically all of these sequences are valid combinations of the same underlying base structure, because they all have almost the identical structure. If you align them, they're essentially probably less than 5 angstrom RMSD from each other at most. So this is kind of like why we want to explore the sequence space. But the problem is I can't ask Chad to test 1,000 proteins for me. So that's why we had to pick 60. Okay, so the first part is, if we remember, the whole pipeline was we got to start with the MSA and we got to give it some assigned labels. So to give it those assigned labels, we're going to have to do some molecular modeling and predicting the structures to get some structural feature that correlates and wants to be the label or is the label we want to train on. So in this particular case, AlphaFold is really great. There's a notebook. So I guess every slide is formatted the same way. There's a code snippets that I think if you scroll in the notebook and find are kind of like the main points of that notebook, that you might just want to look at that cell if you're interested in, if you don't want to run the whole thing. And then I also have like the URL at the bottom. So I'm gonna also upload the PDF to the repo later. So if you don't have to like worry about something, you just have to remember one link. I guess the GitHub repo to like Charlie's lab or this particular paper, you don't have to memorize all these paths or jot them down if you need to. But essentially, AlphaFold is really great, except if anyone's had experience running AlphaFold, it takes a really long time. And it's because the AlphaFold is 2 parts. There is an MSA generation and template search on gigantic 2 terabyte databases. There's a smaller version, but it takes like several hours for like a 400 amino acid protein because it's all CPU based. It's running Jackhammer, like, or HH Blitz or something on a large data set. So all these sequences are basically almost identical. I mean, they're 40% identical. So most of the time they're going to have the same hits. So it's kind of wasteful running the inference pipeline and searching the entire 2 terabyte data set. So that's the point of this notebook. So what I did was, remember how I said all of these proteins have the same underlying backbone? They're kind of just evolved to have different variations, but from the same template. Is we actually we could build that template. We can get the consensus sequence, which is kind of like the average sequence. If you took the 800, took the average across the columns, that's what it is. And you can, so I'm doing that with HH Consensus, which is a tool from HH Suite, which is a really awesome. I think a lot of bioinformaticians use HH Suite. It's pretty great for fast sequence alignment. Um, but yeah, so we built the consensus, and then on my computer cluster, I had enough resources to run the full inference pipeline with the full database. So I ran that, and the output of that run is stored on the repo. So everything you need to do to reproduce any particular thing, you don't have to download anything. Most of the notebooks should be just run with the data in the repo. So for example, this one, there's a directory right in the same example folder, and it holds the results for the consensus run. And then we can actually, if you're interested in what the output of AlphaFold looks like, I would recommend looking in that directory if you're interested, because I take the MSAs that were generated from that run. So AlphaFold has three different MSAs that it generates and then kind of combines them together. So before they got combined together, I just took it, put it in like a giant list of FASTA sequences, and then I'm saying, this is my new database. It's not as big as the 2 terabyte one. It's about a few megabytes. So yeah. So it takes about like a few, maybe like a minute to run on. So and now talking about actually running AlphaFold, so that's the second notebook. So we have this data set of sequences that we're going to use for our new MSA inference pipeline. So what happens is instead of the three data sets or the three MSAs that AlphaFold, the traditional AlphaFold uses, I just stole their code for Jackhammer for one of the Jackhammer inference or MSA generation steps, and just run it on my particular data set. So if you so it's pretty modular. So if you look at the actual inference code for AlphaFold, it's one script with like 400 lines, and a lot of the paths don't make any sense. Or like there's like 20 paths that you have to set just to run AlphaFold. So like it's really nice that you could just run it from this cell, and then any particular parts you can look at this function, and then this function will say, hey, I'm building an MSA. And then the next step, hey, I'm running inference on a GPU. And then, hey, now I'm relaxing in Amber. And all of those are separated out as individual steps. It actually makes it a lot easier to learn how AlphaFold works, because I had to go through the original script and convert it to this, and it wasn't the easiest. So I mean, yeah. But for like context, because I think a lot of you are, how many of you are computational? Raise your hand. Okay, we got a handful. So you might have access to like a GPU that you can run your Jupyter server on, and have a GPU available to your notebook. And so you can run the MSA generation with the CPU, and it's gonna throw out an error. Hey, I couldn't find a GPU. So But if you have a GPU, you can plug it in or launch Jupyter Lab from that node with the GPU on your cluster or whatever. It takes like 5 to 10 minutes to run this example. So the nice thing about all these notebooks, so sometimes people release training code or notebooks, but then to actually run it is kind of a hassle in itself. It's like, hey, do I really want to set up distributed training with all these GPUs to train them on? Like, no, I just want to read the code and understand what it's doing. So all of these examples, all the notebooks should run under 10 minutes, except there's one notebook for training the random forest models, and that takes like an hour, but it's not that bad. So ideally, you can look at multiple notebooks in the course of a day, so you can kind of understand the pipeline. And if errors are thrown because you don't have a GPU, you could just skip to the next notebook. None of them are reliant on the output of the previous one, so you could jump in anywhere you want. Like if you don't care about the consensus sequence, I won't blame you. It's not as interesting as AlphaFold, so I would just go to this notebook. Um, so the next thing is, okay, can you really just take away the three different MSAs and 2 terabytes of information and say the consensus sequence hits do as well. So the answer is I think so, because if we look at, there's no actual real way to validate this. I can't make like 1,000 crystal structures and align them. But what the nice thing about AlphaFold and most modern, like AlphaFold 3 and ESM Fold and Rosetta Fold, they all output confidences because people love to be confident in their predictions. They're like, I got this structure from AlphaFold. It was really hard. Do I use it now? And then the great thing is each residue is scored by the quality from a confidence block in AlphaFold, which is pretty cool. So if we look at the average predicted, I forget what LDTD stands for, but it's some representation of sidechain environments and making sure that the environment that the residues see in one protein is pretty similar to the environment in the other. Because if you do simple backbone alignment, might not capture the perfect RMSD type of thing. So their predicted score goes to a max of 100. All of mine are mostly above 85, which I would say the cutoff for a decent structure, I would say is 80. So that's pretty reasonable. This is probably what you would see. You know, most of them are good. Some of them are stragglers. If you're interested in what the stragglers look like, I do have in the SI, I do talk about one straggler and color it by the PLDT. And you can see like the central core is colored in blue and it's like super confident. And then all of the loops and everything near the surface is red, which is typically what happens because we have the most sequence and template information for like the central core. And you could still use that for modeling because I'm not docking like the outside region. So like that's like kind of an example how to because half the battle is understanding how to use this thing. So even if you get low quality structures, you still need to look and say, hey, is this in part of the protein that doesn't really matter. I don't care about this loop is inaccurately modeled. So if you're interested in that type of exploration, I recommend that. So next thing, so AlphaFold 2, and I guess the current version of AlphaFold 2 is 2.3, which is really good. It's really good for monomers, and that's about it. So AlphaFold 3 came out and it does, small molecules, nucleic acids, cofactors. It's really great, but they were distracted by the pursuit of money, so it's not open source. Okay, I mean they have a nice server, but like you can't predict thousands of structures on demand for any analysis you want to do by like pinging their server or running some script to scrape the web page and make inputs for you. Um, so instead of that, I kind of re-implemented something that also came out around the same time, which is AlphaFill. So AlphaFill is also a web server available with a lot of pre-generated AlphaFold structures with cofactors. So I definitely would check that out. They do the same thing. They basically look for the PDB, align it to a structure, transplant the cofactor, and then minimize it in a force field. So I do the exact same thing here. I'm just using a different force field because Charlie is one of the biggest developers of Charm. So Charm is a molecular mechanics package. So basically, if you know any, it's like Amber, OpenMM, Charm, Bromax, anything for simulating proteins for like, you know, I guess long time scales on like supercomputers. That's the type of I guess tool it is. Charm's really great, but it was written in Fortran, which sucks. So something cool that we're working on. So OpenMM has a Python interface, is really nice. Our lab is also working on a kind of interface for Charm in Python. So you could see like, hey, I could do some pretty cool stuff with some Python looking commands, like set up absolute harmonic restraints on the protein backbone with a specific force constant. So what that means is you're setting up springs on each atom in the protein to kind of hold it still because we kind of want to minimize the cofactor into the protein. There's previous steps for minimizing side chains, but the main step is putting the cofactor in there. So then what we do is we minimize and run. So there's two types of minimizations that are commonly seen in charm. We have steepest descent, which is just really good for just spitting out an answer really fast. And then there's a fancier minimization called adopted basis Newton-Raphson. I don't know, but it's a slightly fancier optimization method that calculates a few more derivatives, or I don't know, like and so basically that's all pretty cool, and you could do it in a cell. And then same way, AlphaFold spits out a confidence value. This doesn't have a confidence value. Molecular mechanics force fields, what they try to do is spit out an energy term that you can basically, or it's a potential that you can differentiate to get forces to move your protein around. So it already comes with an energy function. So we could just look at the interaction energy between in vacuum between the AlphaFold 2 structures and the F80 cofactor. And the red line is like the interaction energy I got from a crystal structure. So most of them are pretty on par and actually tighter binders to F80 according to the APO prediction from AlphaFold, which might not necessarily be true, but it's not bad to see this. So that's great to see. So if you're interested specifically, because there is a really huge barrier to entry with molecular mechanics, because it's kind of like you have machine learning, and then molecular mechanics is kind of doing its own thing, and most people don't use them together. Like AlphaFold, giant machine learning pipeline. At the end, they just minimize an amber. And there's the amber minimization, and that's all they use it for. But molecular mechanics is a lot more than just minimization in basic MD. It's about like force fields, it's about forces and docking and prediction of binding poses and sampling dynamics of proteins. And it's pretty good at that. And it's been good at that for 50 years. So if you're interested in that, that's kind of a dive in its own. But there is a really good PyCharm workshop repo in the Brooks Research Group repo. So if you really want to get into like, hey, I want to do free energy methods, you can jump into there. But this is kind of like, pretend like this is the iceberg. Minimization is just like, what you can see, and then there's all the other stuff you can do. So yeah, so another really cool thing, we also have our own docking tool in Charm, called FFT dock. It's a pretty fast rigid receptor docking method. So I guess for context, dip dock, the inference time is like 40 seconds, and the performance on their training spit in a PDB bind was like 40%, less than 2 angstrom RMSD for the first pose, something like that, which isn't bad, but we're interested in stereochemistry and activity, and DiffDock doesn't really care if my substrate has a hydroxyl group sticking in a different part of the structure, because it's kind of just trained on a bunch of just data to spit out a model. And it might learn that stereochemistry is important, but like a force field will definitely tell you, hey, the ligand doesn't sit that way because it's on top of some atoms and the energies are crazy. So that's why, um, I think if you look at the paper for FFT doc, it's like 60-70% performance for using the known ligand conformer in the hollow structure, which is is decent. So, but obviously that's not the apo structure. That we don't know the true ligand bound pose. So the point of this script is just to pick a random conformer, do fast rigid receptor docking, generate some initial placements for some downstream minimization in the next script, so we're going to minimize it in the force field so we can get like rescored poses. And then so you can run that if you're interested. Also on CPU, the FFT dock example requires a GPU. So you can see here number of GPU devices. I'm using a 1080, so it doesn't require a lot of resources because the 1080 is pretty old. Yeah, and what FFT dock actually does, it basically spins around the conformer you give it in not every possible, but like finite number of different quaternions that represent different rotations around a sphere. So the number of orientations in this particular quaternion file is 36,000, which is, I guess, a lot. So that's a lot of poses. And you're like, okay, how can you score all of them really quickly with the molecular mechanics force field? So that's where the GPU part comes in. It uses the QFFT library from CUDA, and it uses grids of the protein. So you can just grids of the protein and ligand. And one thing you can do with grids, or you can convolve them really fast and in parallel. So you can evaluate each of those possible different ways to convolve that image as a, yeah, I guess, yeah, that's something you can use. You can use it to find matching part of an image really fast. So this is the same thing. We're just putting an approximate grid potential that kind of represents the full atom force field, but not really. So it's kind of like a rough, fast placement. But it takes about, I would say it takes about 5 seconds. The grid generation takes a little longer, but this is actually really fast. So it's a really nice tool to have. The problem is it's just a little difficult to set up because Charm by itself is difficult to run. So I would just look at this, get inspired by this, and say, hey, there's tons of molecular mechanics docking tools. Like, Autodog Mina is great, and outperforms DiffDock. But, maybe I should look into that when you're thinking about which docking tool you want to use for modeling your system. So the next thing is, yeah, so FFT-doc, we're basically convolving grid representations of the electrostatic and Leonard-Jones potentials. So you don't get all the fancy charm force field terms like dihedral angles and side chain torsions and bonded terms. So we can get that by just taking those poses and minimizing it, because the hard part in docking is kind of the generative problem, because Cartesian space is huge. So this is kind of like a workaround. Since we already know the binding site, we could just explore a sphere centered on, like, well, it's not a sphere. You just slide the ligand across the entire grid that you choose is like the bounding box of your binding site. And then we take those initial guesses for the energies, pick the best 500, and then minimize those top 500. So it's kind of like rescoring them. So if you see a paper, like there's a lot of papers coming out about, hey, we use Autodoc Vina and we did some consensus machine learning rescoring. And you're like, I don't know what that means. So all that means is you take the predictions from Autodoc, run an ML tool on it to predict the binding affinity, and then use that as part of the score. So this is the same thing. It's not very complicated. It's just evaluating it in a different, with more resolution. So which is what force fields are really great at. So next up, so we have this 500 poses. We're just going to cluster them. So basically any unique ligand pose is represented by a cluster that's like, hey, out of these 500, these were within some RMSD distance cutoff to it. So technically all these different minimized poses kind of represents the same cluster. And if we take like a representative out of it, that representative has this particular score. So we'll just say, hey, the cluster has that score because I'm clustering by 1 angstrom RMSD. So most of the structures, like we can look, the average energy, sometimes FFT doc puts atoms on top of each other, like really close. So, we're only really concerned with, hey, a lot of poses found the same orientation, and if we try to assign that orientation a score, we'll just assign it this score, which is from the best score in that cluster. So that way we can break it down into a different problem. Now we have 500 poses, and it's kind of like an ensemble, and you're like, each pose has a energy and a particular orientation. And now we want to figure out, hey, how can I, so now we can represent unique individual clusters. So instead of 500 poses, now you're dealing with, hey, let me use the top 10 clusters or the top 5 clusters. and do stuff with those poses. So that's why clustering is really useful. The next thing is now we're done with the molecular mechanics part. We did structure prediction. We did adding a cofactor. We did docking with initial placements. We refined it in a force field. And then what we did is cluster them. So now we have some list of top five uh, top five clusters and their associated energy and an associated pose that represents that cluster. So the next thing is for each of those clusters, I'm going to take that representative pose and I'm going to try to figure out, is it sitting, which face is being presented towards the cofactor. So you can mess around with some, distances. So in this script, what it does is you can represent the plane of the ligand. So if we pretend like the ligand docks into the protein and the cofactor is right here. So if you get the normal, or if you get the plane of that ligand, that basically is the face. And then if you get the normal vector, it tells you where's that face pointing. Because if you flip a plane, that makes the normal vector just go the other way. So that's the point of the script is I just happen to pick an arrangement of three atoms on the ligand and generate a plane from that. And the normal vector for this particular plane is pointing this way. And in this particular example, just the way that it is, that happens to be the R face being presented to the F80 cofactor. So that's why in this script, after you kind of label each cluster, so you have a bunch of energies, you have a bunch of counts, and you have a bunch of labels. And there's like a fancy thing in StatMech, which is like macro states. So you have micro states, and each micro state is the 500 different poses. The macro state is the cluster, and then you can figure out the kind of relative probability of each cluster. So that's the whole point. You could honestly just take the top one cluster. This is just in cases where the energy difference is really close. So if you take the top one, it's gonna give you, maybe it's R. And the other cluster is like maybe like a kcal per or like half kcal per mole less. And it does S. So technically you have evidence that this enzyme is racemic, and maybe you should label it as 50-50. So that way, if you do the macro state stuff, you can assign based off the energy differences, the probabilities to each macro state and say, hey, all of the R macro states have these probabilities, all of the S macro states have these probabilities. What's the probability of it being R or S? And then in this particular case, this is tropy. The reason that this sequence or structure is always being modeled in all these scripts is because if you read about most of the work between the Brooks and Narayan lab about these enzymes, tropy is the most common one that we study because we have crystal structure, we've done flexible docking, we've done QMMM. So there's tons of stuff you can read about and compare with like the actual known bound pose if you're interested in seeing how well docking tools recapitulate kind of the final bound pose without further molecular dynamics. But in this particular case, because we knew the binding site, we rescored the top poses with some information that we have about the system. We get a pretty reasonable accuracy in labeling the stereochemistry of each of these enzymes, because if you think about it, even before we do any analysis, so we know that we have 60 enzymes, and there is a switch from one mutation, phenylalanine to tyrosine. And you might be like, that should be pretty easy to capture with the molecular mechanics force field. It's a switch of one atom. You go from hydrogen bonding, you have a huge electrostatic interaction between the anion of the substrate, which is not neutral. It's actually negative at this pH. So actually strongly interacts with that one residue. So it's kind of pretty obvious that if you take a bunch of structures and most of those structures just have that one atom difference and all the other residues are different, that you should see a pattern emerge naturally from the force field. So, you know, if we just focus on those 60 enzymes that we have the most information about, those 60 enzymes are sitting on the bottom here. And each row is numbered by a bold number, and the bold number represents a different substrate. Most of the time, these enzymes are the same across the substrate. So, like, for example, this enzyme AZA-H is one of the exit enzymes. TROP-B is the one that's well modeled. I guess it's supposed to be R, but I guess it's dead here. But essentially, AZA-H, this enzyme does the blue box with each ligand. And then you might ask, well, what is this blue box? Why are they boxes? Well, blue means it does R. That's what Chad saw from the cell lysate. And then a gray box means it was dead. And a yellow box means it did S. And there's some green boxes here that mean racemic. We're just going to think about this as a binary problem because it doesn't really appear that often. So this is the data that Chad saw in his paper. And based off this data, he was able to make some conclusions and prove that a particular residue flips the stereochemistry and is the dominant residue that controls stereochemistry. Oh, oh, okay, cool. Oh, dang. All right, all right, we'll start skipping around. Okay, so basically, if you read the paper, if you align all these squares, it's about 70% accurate. So, but I have them in the SI, there's like lots of stuff about that, like different accuracies for different docking parameters, and you can mess around with stuff. But then the nice thing is, hey, because it's high throughput, well, let's just fill in everything. And instead of just 60, why don't we fill in all 1,000? And that's what you could do. I'm going to skip reactivity. So you can also do the same assign label for reactivity. That was all for assigning the stereochemistry label as basically 0 or 1. Okay, so basically, if we skip reactivity, so this one is for generating the MSA for training the machine learning model. This next one is taking that MSA, taking all of those R or S labels predicted from the docking results, and training a model to map 800 sequences represented by 1 to 200 MSA columns, because after filtering out the ones that have gaps and are kind of not in the binding site or second sphere, that's kind of the data set that you're working with. And take those 200 columns, predict if it does S or R. And this is a little bit, you might be interested in this because this is an auto ML implementation. So what it does is it actually runs like 30 models for you at the same time. It takes like an hour. So it's really cool. I would check it out. And then we could do SHAP. And then, you know, if you dive into SHAP, there's dependence plots. So this particular feature is the column that represents the residue that controls that stereochemistry with that one atom change between phenylalanine and tyrosine. So the nice thing is I could just send this plot to Chad and be like, hey, it looks like phenylalanine negative means it's more S because I set S to -1 because it was -1 for S, 0 for racemic, 1 for R. So negative means S, positive means R. And it's pretty easy to see like, hey, maybe you should mutate that one residue. And you can do that for a lot of residues. And you can just label how important they are because SHAP tells you how important each feature is. And you can make any of these plots. So these are the labeled residues here. There's some discussion in the paper how they actually relate to experimental residues. And we could do the same thing. We could predict which ones are important for reactivity. Some of them overlap, some of them are different. One important, like, so an example of how you can use this, tell an experimental collaborator something to try is like, hey, we can look at M54. We could generate this figure. We could send it to him, ask, did it do anything? If it did, then we discovered a novel residue for controlling the reactivity of an enzyme. And then we could do that for 10, or we could do it for 100. The nice thing is you could do it for as much as you want because You could just add more sequences. You could predict mutations. You can plug in any variant you want and predict the structure of it and slide it in with the rest of the sequences in the MSA. So it's a really useful prediction tool as well. Okay, so I guess, yeah, perfect time. So kind of that is the main summary of the first part was just concepts you might be interested in or heard of if you're looking in this like particular field, because it's a really fast changing field. Because it was, if you think about it, 2022 AlphaFold, and then we have DiffDoc, then we have AlphaFold 3, then we have CollabFold. There's like so many things going on. It's really cool. So I recommend just perusing, like just looking at these words. So when you read a paper in the future, you can be like, yeah, I know what docking does. I know what rescoring in a force field does. And so I think that's the great part is you could just look at the code, get the concepts, and have a better understanding of what these tools actually look like, which I think is a big barrier in kind of science communication is like every time they present AlphaFold, it's this super high level concepts, but we kind of just want to know how do I run it and why do I care? And that's the point. Each slide was about why you could care about any of those particular fields. So I guess I don't have time for this, but this is in kind of a collaboration with Chad and the Narayan group. I'm actually testing some mutations experimentally from this pipeline, and we could see like basically I tested some experimentally in cell lysate and found a bunch of mutations that, so they're colored green. So all these green ones here are new and in the second sphere. So traditional approaches have a really hard time going outside the binding site. So it's nice that I have some initial experimental evidence because, for example, these mutations make this enzyme more active towards R. Same thing for this guy. This guy makes it slightly less activity, but way more selective. And this one revives activity. So there's a set of pretty cool mutations. I will have to spend several months purifying the enzymes. So that's why it's in preparation. But I think it's really cool that there's some evidence that all the stuff I did, it doesn't have to be just computational. It could lead to some experimental collaboration. You could steal any part that you want. And so yeah, that's the summary. So the goal of this talk was to talk about tools and technology in high throughput machine learning and molecular mechanics for protein modeling. Um, Current approaches are making modeling more and more accessible. So nowadays, I think all of us should be at least somewhat familiar with, I can, go on AlphaFold 3's website and predict the structure. And then there's just a whole spectrum, sorry, there's a whole spectrum beyond that, and this is part of the spectrum. So the goal of this paper was talking about how we can combine all of these different approaches, make some tiny adjustments to them because of the domain knowledge of the system I have, but adjustments that you can make based off domain knowledge of the protein system that you're studying. Because each part is from a generalized technique that has like pretty rigorous, so it's based on force fields, which have a 50-year history. Docking has like a 30-year history. Structure prediction, how many, it's like the 15th CAS competition going on. So none of these are like crazy deviations from what the central concepts are in each of these different tools. And yeah, everything's open access, the code, data, paper. You don't even need to log in to read the paper. It's open access. So yeah, acknowledgements. This is a really fun project. I need to acknowledge my PI because he was the main reason why I did all this stuff. He kept asking, why are you doing that? And I'm like, it's a good question. And then Chad, because the whole reason this started was because Chad did that original ancestral work and asked me, hey, can you model one of these structures for me and help me make a figure? I was like, yeah, and it turned into this. So that was fun. But I think that's kind of like the PhD experience is you join a lab, your PI tells you in your first year, hey, you want to try this project? And then 5 years later, you're still working on it. So I think it's a common experience. And then a big shout out to Allison because I've basically been working in her lab for a year, making all these mutations. And then Troy for the original ancestral sequence construction work. Um, But yeah, like thank you for your time. I think this was oh okay.
So we are over time already, so I think we'll just need to take any questions offline.
Yeah. If like is this room being used for anything?
I'm not sure if it's
Okay.
possible.
Okay. I
We can
mean,
have it for a little bit longer, so if
yeah.
you're available to stay and people want to
Yeah,
stay.
I mean if there's anyone who like wants to try something and since I'm here it would be easier, you can ask. If not, then the slides will be a PDF online. So and there'll be a video, so it should be accessible whenever you want.